Today, I just want to go over some of the basics related to complement proteins. Complement proteins. These are not proteins that say, you look nice today. Sorry, bad joke. All right, complement proteins. The complement system is a series of about 35 proteins that are inactive all the time in the bloodstream. So 35 inactive proteins constantly circulating around the body, waiting for the right chemical cues in order to activate. So the complement system then is these 35 inactive proteins circulating in the blood system, and they activate and assist in the destruction of pathogens. And this assistance in destroying pathogens is where the word complement comes from. Assistance, complement. So when the right precursors are there biochemically, they activate and they assist in the destruction of pathogens. When these right precursors are there, a complement cascade begins. And this basically is a biochemical reaction where many of the complement proteins coat the microbe in order to disrupt the cell membrane. Phagocytosis is what we talk about, where the cell membrane is broken and then the cell itself is consumed or eaten. So they help, the proteins help in the disruption of the cell membrane, eating through it so that the pathogen might be consumed. It's an assistant to the immune system in essence. So basically there's three ways that the complement proteins work in the body, three activators. The first one is called classical complement cascade, and this activates when these proteins come into contact with an antibody that is attached to an antigen-antibody complex. So it gets the signal chemically when an antibody is attached to a pathogen, and the protein then in the blood initiates this complement cascade and starts to coat that area and assist the antibody in killing that pathogen. So the trigger, if you will, was the presence of the antibody, and they're like, oh, hey, here's the antibody, and they glom on, activate, and assist that antibody in destroying the pathogen. The second way that they work is called alternative complement cascade, and this occurs when the protein is cued to detect a pathogen on its own. So there's no antibody actually on the pathogen yet, and there are some infections that don't involve antibodies. So you've got an infection that doesn't have an antibody, the antibody's not there to create a classical complement cascade, but the protein still picks up, this particular protein picks up the chemical markers and says that's a pathogen, and then they come out and attack and surround and start the coating process to disrupt the cell membrane. So this is alternative complement cascade. And then finally, there's the mannan-binding lectin cascade, the mannan-binding lectin complement cascade, which activates when there is carbohydrates, certain types of sugars found on the surfaces of certain pathogens. And so they detect the presence of that particular type of carbohydrate or sugar there on the surface of the pathogen and realize, this is what we were looking for, and then they attack that. So the protein binds to the pathogen because of the presence of the carbohydrate on the surface of that pathogen. So just to recap briefly, the complement protein system is a series of approximately 35 proteins that remain inactive in the blood until certain chemical markers are present. And when those markers kick in, it activates and assists or complements in the destruction of pathogens. When those biochemical markers are present, a complement cascade ensues, where the complement proteins coat the microbe in order to disrupt the cell membrane and assist the destruction of that cell. The classical complement cascade is when an antibody attached to a pathogen is detected. The proteins then assist that antibody. The alternative complement cascade is when there's a direct pathogen, an infection that does not have antibodies. Antibodies are not involved in that particular type of infection or pathogen, but the chemical cues are still there. The proteins then come and grab onto that. And then finally, the mannan-binding lectin complement cascade, which involves the carbohydrates or sugars of a particular type on the outside of the pathogen that the proteins then cue to, coat, and help in its destruction. So basically, this is just a brief overview of the complement proteins and the complement cascade system that our bodies use to protect us. If you'd like to learn more about this and related things, beneath this video you'll find a link. If you'll click on it, it'll take you to that website where you can get that information. And while you're there, on that website, you'll also find a link to an ebook that's ready for immediate download.